Okay, we'll get started. So last class we just had an overview of what we are going to talk about. Today's class will focus on a very short introduction to molecular biology. This is lecture 2A. There's a lecture 2B which we'll follow. Again, the class today will go till 6:25ish, where the idea will be in the first part of the course. The first part of lecture 2 is to cover some concepts overall to give you a flavor of what is molecular biology. And the lecture 2B is where we'll start to do some computational exercises, very simple probability and stats, but you'll start to see the flavor of how biology and math mix in. Okay, any questions? There'll be questions. So for some of you, I've already approved the registration. For those of you I have not, there is a reason I have not, except for one exception, uh, because I either found the motivation to be not very strong or it was a bit over the place. And I'm very serious. We just want people who are actually motivated to be in the class. If you're taking it just for the sake of, just um, as a function where you're not, you just want to take a course in digital health. Unfortunately, this course is not for, is not for that. It's only for we come here, we have fun, and you go back and still have more fun. We're not here in the regime of bringing more numbers. I don't play that game at all. So we'll, if you have not, if your registration has not been approved, please email me. Unfortunately, after today, I will not have, after today's class, I will not have time to spend. I have to rush. But please feel free to email me to explain your motivation. And if I find it convincing, I am happy to approve it. But the reason it's not been approved is there must be a reason that it's not been approved. Okay. Okay. So the first thing that we're going to talk about is what is a gene and what is a genome. So a gene is nothing but the basic hereditary unit of hereditary information in any living organism. Broadly, it is used to classify when a particular thing will make protein. So you can synonymously understand genes to be a protein. It's something that's going to get inherited. We'll talk about how it gets inherited and what part of it gets inherited and so on in future classes. But broadly, a gene is something, it's a functional unit of the DNA that will make protein, and this is essentially what gets conserved or remains conserved across different cycles of progeny. There's another word called genome, which is the complete set of genes that encode the genetic information, right? So there is gene, which is a unit in itself, and then there is genomes, which is a complete set of genes that encode the genetic information. We have around, overall, we have around a trillion cells, and each of these trillion cells will have DNA, but that DNA does not exist on its own. It's wrapped around these different proteins, which we'll talk about once the time comes. But broadly, we have 23 pairs of chromosomes. 22 of these are what are called autosomes, and one which is the sex chromosome, right? We have two copies of each of this. That's what this implies. There is partially one copy of it broadly is coming from your mother's side, the other half of it is coming from your father's side. Again, it's not as simple as I'm making it sound right now, but for this class we will assume that 50-50 is what it comes from. Autosomes again are your chromosome. So we have so usually these are numbered, right? And then depending on a male versus a female, you will either have X or Y. How are they numbered? Any ideas how? Why is one numbered the one? It's actually the largest. Okay. So it goes in this direction. You can actually see it here as well. It starts from a very long thing and then it goes down all the way till it comes to this pair. So females have this. I guess you might know, what do females look like? There are two copies. So everything else is shared by both males and females. So there'll be 2 copies of each of this, which is what you see there. And then there is a chromosome XY or a chromosome XX, right? So females are XX and females are XY. An important factor here to know is this is sex or gender? This is sex. It's a biological phenomena. Gender is something that you identify with, but sex is biological. It depends on what kind of chromosome structure you have. If you're an XX, you have, or if you have more, basically if you have more than 1 copies of X, you are towards the female spectrum, right? And otherwise, you are a male. Now, you can think what kind of manifestations this would have, right? These are your chromosomes. So, for example, if a chromosome looks like this, there are certain portions here which form your gene. And we'll talk about it in just a bit. This gene will give rise to a structure like this, which is, my drawing of the protein. If there are 2 copies of this, we have enough mechanisms to produce essentially a lot of protein because there are 2 copies of the DNA to begin with. But that can be problematic, right? In this case, because in females, I have 2 copies, whereas in males, I have only 1 copy of X and 1 copy of Y. Usually, in most cases, you'll not see much going on in this gene. Or codes for proteins, but not very, it's not the most densely populated gene. What is, however, required is you need this particular X copy to be present. There are certain proteins that get synthesized here, but you have to evolve a mechanism through which you can silence 1 copy of this chromosome. The reason I'm mentioning all of this, and this is not on the slide and it will become clear a couple of slides later, is remember that this is the definition of being a male or a female. Okay, so everyone on the same page so far? Okay, we have besides this genome that we're talking about, which resides in the nucleus. It is there on the next slide as well. There are other there is one particular other set of chromosomes or genome which is called the mitochondrial genome, and you essentially inherit your mitochondria from your mother, right? So you essentially gets passed on from your mother. This also has around 8,000. It's, while we have 2 copies here, we have actually multiple copies of the mitochondrial genome. It has its own mechanism of copying, making proteins, making RNA, and so on. But for this part of this class, we will focus mostly on the DNA. At some point, we'll start to venture into what information we get from the mitochondria, how does it play a role over on the cell. But for now, what you should understand is we're going to focus on the nuclear part, but there is some genome residing in the periphery or in the cytoplasm, which right now we are not going to talk about. Okay, this sequence of this nuclear genome, I can in principle calculate how many base pair it is made up of, right? So all of this is 80 GC. I can calculate the length of this, which you can also see on this what's called the karyogram. This is an image, literally an image of a cell which has been burst, which is a nucleus which has been burst open, or you can actually put your microscope there and see what it looks like. Often you can have multiple copies. This is called trisomy, right, which have one extra copy of one particular chromosome. In this case, it happens to be 21. Usually, whenever this happens, there is something wrong that's going to happen to the person. You will get one disease at some point in life. Okay. But to come back, if I calculate the length of all these, by length I mean this is 1, this is 2, and let's say this is 100. If I do this exercise for all the chromosomes, I get 3 billion base pairs of sequences. So if I want to know the sequence, you have 3 billion base pairs, I have 3 billion base pairs. That 0.01% is what makes you you and what makes me me. Let's talk about this field called genomics and what is multiomics. So as I mentioned in the last time, in the last slide, the genome itself, does not exist on its own. The chromosome itself will not survive on its own. It wraps around these bodies which are called nucleosomes. Once we start to talk about epigenomics, we will talk about what role exactly do they play. But this brings in a lot of stability to how the DNA is packed. It wraps around this nucleosome 1.65 times. So each nucleosome will have 1.65 of its DNA wrapped around it. So it's literally like a string on a bead kind of a model. This helps compaction overall, so it can allow the entire set of chromosome to be very, very compact. If you were to, how much will it cost if I were to open your cell and untie this entire chromosome structure? How much do you think, how lengthy will it be? Okay, any other guesses? 600 billion kilometers. It's often quoted like that in the media, but I don't think that's correct. Anyone? Length of DNA? So there are figures like that, which is, it says that you will go all the way from Earth to this. I think it's a few meters. I don't think it's that large. Okay. Anyway, so we'll not worry about that. But broadly, what you should worry about, at some point, it will start to become clear, is depending on how this wraps around, you can allow access to other parts of the genome. So if I compact it, I'm closing my entire DNA. It will not become accessible to other things. Whereas in this case, if there is not much wrapping around happening, which means that nucleosomes are too far apart, this part is where genes usually sit. And you need this to be open for the genes to start producing RNA and then protein. Okay. So this is what's broadly called open chromatin, which is where your transcription is active, and then there is something called closed chromatin where transcription is reversed, right? The reason to include this in the part of the slide, the first slide, before actually I introduce what is transcription and translation, is because I'm going to describe what is genomics, right? So the genome essentially is this part of DNA along with these proteins, which are called histones or nucleosomes. So each nucleosome is an octamer. It is made up of 4 different histones. You might have taken BB101 or a simple or a similar course, so you might know what these essentially bodies of proteins are. Okay, any questions here? Okay. Okay, so the reason this course is called multi-omics is because it tries to study multiple of these omics technologies. So let's talk about how did this word come about to be. Right, so we already mentioned that gene is different from a genome. Genome is the entire complete set of instructions required, which is essentially a mix of all the DNA plus all the extra stuff that is required to stabilize it. This word itself was coined in 1920 by a botanist, Hans Winkler. He writes, I propose the expression genome for the haploid chromosome set, right? So he's very, very specific. It's the haploid chromosome set, which together with the pertinent protoplasms specifies the material foundation of the species, right? So this is the complete set of instructions, the haploid set. So haploid is 1 copy, diploid is the second copy, or the 2 copies together. Uh, this is often, assumed that genome is made up of fusion of 2 words, gene and ome. Gene is, very, very simple to understand. And, if you're coming from one particular group, you will try to induce certain connotations to this word ome, but it's not that ome that you're thinking about. It's an ome that comes from terminology of botany, where this has been essentially used in the past to talk about a Greek word which is oma, which finds its way into in other fields like rhizomes and so on, which existed even before this particular term was coined, which essentially is an expression for which encompasses all the complex systems or a complex composition. The term itself, or genomics, or this particular coining as we know the field today. So there was genome which existed in 1920. What did not exist was genomics. So this was supposed to be name of a journal which never came about to be. In 1986, this was coined by Thomas Roderick, who wanted this to be the name of a journal. Unfortunately, that journal name never came about to be, but the term itself existed. The difference between genetics, which you might have taken a course on or you might have studied about, is geneticists will worry about just one single gene at a time. There are a lot of people, for example, in the institute, which focus on like one gene at a time. The difference between a geneticist and a genomist or a genome scientist is we look at, like, for example, the field that I work in, we are a lot of genome scientists. We look at the entire pool of genes at once rather than looking at one gene at a time. So when biology started, a lot of it was understood that one gene does everything, right? So if I get cancer, it's happening because of one gene. If I get diabetes, it's happening because of one gene. In the last 20, 30 years, we have learned that biology is much harder because it's more often than not, one gene does not cause one disease. One disease is often a function of multiple different genes. You have something to say, I think. Will offend them, right? Yeah. And of course, this will go online sometime. So I will, once somebody tries to look into my history, they will find all this, and the ball will get rolling. No. So geneticists, of course, try to study, of course, how genes are inherited and so on. But the general focus is on one gene at a time, right? I think that part we will agree with. I'm not trying to sort of say trying to say one field is superior over the other. What we, however, know is that one gene hypothesis no longer holds in a variety of cases. Multi-omics is just the study of different types of omics. So if you are studying genes, broadly genomics, when people mention here, is to do with the general understanding is it refers to DNA, but broadly it actually also means transcriptomics. Okay. Essentially, anything that you're trying to study with gene will become genomics. The one exception that is often counted out is proteomics. And the reason will become clear as you go along the course. There are other courses in the institute for learning proteomics. I will not cover proteomics at all. I will never touch proteins. Okay. The reason is will become clear also again very, very soon, is these two fields have extremely high throughput. You can measure things at scale. Proteomics still hasn't arrived in that truest fashion. You can measure maybe 5,000 proteins, 8,000 proteins, maybe 10,000, extremely expensive and extremely noisy. And the kind of methods that are required to sort of work with that kind of noise do not deal with this sequence itself. Okay, so we will do a lot of probabilities and statistics on the sequence that get derived from here. They don't directly go and apply to the protein world. Okay, and it will become clear as we go along in the course. Yeah. No. That's not what I meant. What I meant is when we deal with genomics and transcriptomics, right, we actually work with individual nucleotide level resolution in most cases, or at least a fraction of them. When we deal with proteins, usually you're not sequencing proteins, right? There is no technology. There are some technologies where you can do some sort of sequencing of protein, but in most cases, when you're actually measuring them in the high throughput fashion, you're detecting whether there is a spectra which corresponds to a particular kind of protein or not. But you don't care about the actual sequence that would have come out of that protein. You would usually not care about, does this, for example, if this makes, let's say, amino acid 1 and CCG, this makes amino acid 2, you would just detect whether these two exist as a peptide or not. You will not care about whether there is actually amino acid 1 and amino acid 2. We don't deal with that resolution at which we deal with in DNA and transcriptomics. It has its own statistical challenges. It has how do you detect whether this is coming from spectra 1 or spectra 2, but it doesn't do this yet in terms of looking at individual amino acids. Right? So that's the difference. So that is a world which is altogether different, right? Because then when you start talking about the conformation problem, then that is one more level of complexity as to how do you actually detect the structure of something. That's what part of the lecture will cover, because that's how the science of being able to detect the structure is what made the entire field possible. But in most cases, detecting the structure or getting the structure of a protein is still a hard problem experimentally, and that's why when AlphaFold came out, it was such a big deal, because that is one problem that scientists have been struggling with for a while. And you can develop physics-based models, but it doesn't scale to the level at which you would want to scale. Okay, so a lot of digression, but we'll go back to doing very short introduction to molecular biology. So people who have taken BB 101, what is one fact that you remember about the cell or about how proteins are made? How proteins synthesized? Yeah, so very detailed, right? So not transported to ribosomes, but translated by ribosomes, right? So very good. In your first assignment or assignment 00, you had drawn some arrows, right? Any thoughts about where did those arrows come from? Is there a law in biology that helps you draw those arrows? Central dogma. Okay, what does central dogma say? Okay, yeah, very, very close. That's what you're taught in school. That our DNA gets transported into RNA, and RNA gets translated into protein. We have around 2 to 5 million species on this earth, and all of these species follow the same law, which will come and become clear in few slides. Life originated on Earth around 3.5 to 5 billion years ago. The first Big Bang happened, just to give you a scale of idea, happened around 15 billion years ago, and we as mammals arrived on Earth around 200 to 350-ish or 500-ish upper cap million years ago. Any ideas about how much divergence exists between human and mouse from million years evolution? So if so, all life began as one cell, and that one cell made all other things. So if I were to say that human and let's say whatever this mice, how long would it be before they would have had a common ancestor? Any claims, any ideas? This itself is somewhere here. We are both mammals. So let's say these are mammals and this is others. This itself would be 350 billion years of ancestral evolution. What would this scale would be approximately? Take a guess. Let's ask for chimpanzee. When did we ever share a common ancestor? It's often said that there are there are ancestors, right? That's not correct. At some point we both shared the same ancestor, but we diverged from it. Any ideas about this scale? Chimpanzee, when would we have shared a common ancestor with? Of the order at least. You know, this is entire 350 million years ago, right? So let's say if I tell you this, if this is 100, yeah, so that's close enough, right? So 10 to 15 million years of evolution of ago or ancestral years ago, we had a common ancestor, and then we started having mutations, and then we started to give birth. Uh, so overall, we have we have 2 to 5 million species. This is an underestimate because we have not been able to actually account for a lot of fungi, a lot of other organisms that exist in places that we have not been able to go. Broadly, if you want to understand life, you can understand it on this time scale, right? So we've already talked about the first set of life appeared here on Earth around 5 billion years ago, or 3.5 billion years ago. Broadly, you can divide the life that exists in the world today as prokarya or prokaryotes, archaea, and eukaryotes, right? Or prokaryotes also have another common name, which is a mix of bacteria. Prokaryotes are called so because pro stands for before and karyote stands for nucleus. They have something which is before a nucleus. They don't have a nuclear membrane. They so their DNA sort of exists inside the cell itself. There is no distinction boundary. When I showed you the first slide, I drew humans and I drew the nucleus inside. I drew the DNA inside the nucleus. In this case, there is no boundary. There's no nucleus. It just exists inside the cell. Um, there are 10 to the power 6 to 10 to the power 7 nucleotides, which means there is 500 kilo base pairs. So kilo base pair, kbp, to 10 mega base pairs. Again, there is no, this is what we know about. A lot of these prokaryotes might not have been actually studied so far. So this is what we know. They house around 1,000 to 6,000 genes. Okay. How many genes do humans have? We discussed this. Yeah, 20 to 30,000 depending on how you count, but broadly that number holds around, right? It keeps increasing every year because people start to characterize new proteins and so on. But it can give you a scale, right? The reason to give this numbers, I will not. By the way, this is for your information. I will not ever ask questions about theoretical stuff or something which you will have to memorize, right? This is for you to get a feeling of what are we studying and what is the scale of being studied. I will never ask a question which requires you to memorize anything. Okay, everything will be conceptual, including both midsems and endsems. Okay, so the reason to give this idea is to tell you about that. We are not very special, right? 30,000 genes going from a bacteria to a complex, so-called complex organism, human, just a 5-time multiplication. So it's not huge overall, right? They can also have 6,000 genes. This is what a eukaryotic cell look like. We are eukaryotes. U stands for truly, so we have a true nucleus. Our DNA sits around here. There are a lot of complex structures here. Again, you don't have to remember or memorize any one of them. This is to give you a scale of things that look different. We have a lot more components. Some of it actually came because at some point the bacteria engulfed into this eukaryotic cell, and then it became part of our genome and so on. But broadly, we of course have 6,000 plus genes in general. It will depend on what the eukaryotic cell is coming from, whether it's a mice, it's a yeast, or whatever, wherever it is coming from. So the yeast genome is 16 megabase pairs. If you recall, the last one, which was prokaryote, went only up to 10 megabase pairs. So there's slightly increase in the size of the genome, but not necessarily increase in the total number of genes. And again, we'll see an exercise in the next class where we try to decode it. Human have 3 GBP. So this is gigabase pairs of genome size. And finally, we have plants, which are actually even higher. Okay. Why should plants have higher amount of why should they have the highest length? We are the most complex and we are the most smartest people, right? Or organisms. Okay. 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 Any? Oh, how do you know that? Oh, the C-value paradox. Okay, we'll cover an exercise talking about the C-value paradox in the next when we do the hands-on, which will be Thursday, next Thursday. Okay. Okay. So, what is the central dogma? Coming back to central dogma, you now understand what is prokaryote, what is eukaryote. You know the genome sizes. You know they are orders of magnitude, can be orders of magnitude different, 10 megabase pair to gigabase pairs in eukaryotes. What is the central dogma? Which is what we discussed just now, which is to say that there is one rule that kind of combines everything, right? Genes are changing, DNA is changing, but one rule that kind of rules them all, because the nucleus is also no there, but you can still make proteins. And that rule is of central dogma. What is dogma? What is dogma? Central dogma. Very good, right? That's the exact definition of dogma, which is what we understand. It's not questionable, right? It's a rule of God, or rule of some higher authority, which is whatever God means, but a rule of some central authority, which tells you, which cannot be defied. It's set in stone. So in your class, you would probably have studied this version of what it looks like, because that's what 1965 Watson, when he wrote his book, the famous book, which still is in print. DNA gets transcribed into RNA, RNA gets translated into protein. DNA can also, of course, be copied. It can, there are ways to copy DNA itself. But often, this is the first version when he wrote it in his Molecular Biology of the Cell 1965 book. But at some point, he will go back and revise it. I think it's the latest version, or the last few versions have been revised to say there were some arrows missing. Right? One such arrow was, of course, RNA to RNA. But this version that he wrote in his book, and which is often misconstrued as the central dogma of molecular biology, is actually not the central dogma of molecular biology. Right? In his 1953 paper, Crick, Francis Crick, the other sort of part of the famous duo or trio, Watson, Crick, and Franklin, talked about this particular diagram, where he very clearly writes that this information, the arrows that are being drawn, is a flow of information. The information here is the information encoded in the nucleotide, which specifies the protein. But what is not possible among all the different arrows, right? So if you remember, this is your assignment 1. Among all the possible arrows, this is all the possible arrows, only some of them are realizable. What is realizable is, of course, DNA can go to DNA, DNA can go to protein, RNA can go to protein, but once your information flows here, I cannot go back. Okay, and hopefully this will also make sense why doing this sequencing, trying to figure out if I can sequence amino acids to figure out what particular nucleotides it came from, is a hard problem. I will explain why it's a hard problem in just a bit. Okay, but is this version clear? This is the central dogma of molecular biology. The realized directions are, in this case, are solid, right, which are general transfers. They happen generally throughout life, but biology also has certain exceptions or specialized transfers where this happens, where your RNA can be reverse transcribed to DNA, which happens in a case of viruses. Your DNA can be translated directly, can have a direct transfer into proteins, again happens in certain specialized viruses. But once you're here, you will never be able to go back to DNA. There will be no information transfer that can happen from protein to DNA or protein to RNA. So when all of this was being developed, right, like the era of 1920s, 30s, 40s, 50s, people did not have this distinction about being a biologist or being a physicist. Everybody used to work on all these problems, right? So Crick, for example, was but what was his actual training in? Crick's training, original training is, so a lot of them actually came from physics, right? Because it became a common ground for a lot of them to come for problems and think analytically. And a lot of these solutions actually, again, I'll show one more such example where it become clear how There were also mathematicians who were answering this question. So I'll strongly recommend reading his book, where he writes about why did he choose to call this a dogma. I forget the name of the book, but if you click on this link, you'll find it. I think it's called My Life in Science or something similar. I think it's called My Life in Science, which a lot of scientists have written, and one version of it is his. So while what you said, which is a belief that cannot be doubted, is what often it is written as, what he wanted to imply was, in a we have not seen, in some sense, the other possibility of protein getting transcribed into RNA or getting translated into DNA so far has not been observed, right? So he did not mean it dogma as in something which cannot be doubted, but given the level of possibilities that could exist, the transfer of information once happens to protein cannot actually makes it back. What he meant to say is to design this as more like a hypothesis, right? So he meant this to help people understand or organize more positive and explicit hypothesis. He actually meant this as a hypothesis, but just because of choice of words, it famously became the central dogma and not like a central hypothesis of biology. Okay. So that first definition, which often a lot of us use, which is DNA makes RNA and RNA makes protein, is actually not what he meant. And he wrote this paper and he coined this term. So we should believe what he implied. And this is, by the way, also he writes it very clearly actually in his paper. It's just that when Watson decided to write the book, it got interpreted in a slightly different fashion. Okay. Okay, why is it dogmatic? Is because of this idea of how the transfer happens, right? And you remember he's coming from this physics background where you talk about information as a measure. So he says that the obvious cases of these have been sort of observed very profusely. They exist everywhere, but there's a special transfer possible, which is RNA, and RNA to DNA and DNA to protein at the present time. The first two of these have only been shown in certain virus infected cells. As far as I know, there is no evidence of the third except in a special cell-free system containing neomycin, though by a trick it could probably be made to happen using neomycin in an intact. So he's very transparent. He's saying at the present time, right? So he's saying that I meant this as a hypothesis, but it became the central dogma, the famous central dogma. Okay. But till this year, we actually have not seen the information flow happen in the other direction from protein to any other words. But again, in a very specialized setting, people have shown that it is possible. But that just shows that this was indeed meant as a hypothesis based on what was observable at that time. Okay, so then became this famous, which was what's called photograph 51. Till I think 4 or 5 years ago, there used to be a show which was also called Photograph 51. I think it still runs in certain parts of the world, which tells the story of how the structure of the DNA was understood. And the reason it was such a central problem to biology is again the problem of science, which is that you can observe cells are replicating. You actually observe that it is the DNA which is a genetic material. We will not talk about that experiment which showed that DNA is indeed the genetic material, because then it will become too molecular biology. But any guesses how would you actually show that DNA is the genetic material? People who have done BB 101, there's a famous experiment which shows it. The mouse. Say that. Mouse had probably not even found their way in testing because it was very complicated. But okay, getting warmer, maybe yes, there is a possibility of doing it with mouse also. How would you share it like something as a genetic material? Okay. 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 Close. Very close. Okay. Okay. Getting even warmer. Yes, correct. But how would you show it? Like, so there are different. So at that point, what you know is, so we'll not discuss the full experiment because it will take time. But there is DNA and there is protein. And I don't know whether this is the genetic material. So that does this get passed on to progeny? So when I mate with this, do I pass on the protein or do I actually pass on the DNA? This is not known. You're engineers, right? Like some of, well, all of you are engineers just because you're in an engineering school. How will you solve this problem? Okay, tag one, huh? Right? Okay, and tag one, which one? Okay, tag DNA. So this is some tag protein. Yeah, you can also tag. You could do some G stuff. Yeah, that's it, right? You'll visualize once you crossover happens. You see more of this, more of this. That's it. You see more of this tag. That means it's the DNA which got the genetic material, which got passed on. Because you will tag this also in the same fashion. But if it is this which gets borrowed, then you'll see more G. That's essentially what's called the Hershey and Chase. There's a book of this is not a book, but when the molecular biology is taught, and if it is taught in the right way, it will walk you through some of these very wonderful experiments, which shows that biology is not about rote learning. It's actually about logic, and this is one such logic. Okay, so what was known at that time is I want to know the structure of the cell, but that is not possible. So we take this DNA, and I know the contribution of adenine to guanine and thymine to cytosine, right? These are, I knew the basis, but I did not know how are they arranged, right? So is DNA like linear? Is it, circular? What is going on that is not, or what is its structure is not known. There was science there to be able to calculate how much of it exists. So what was known by what's called the Chargaff's rule is the percentage of A to T is proportion of A to T is 1, proportion of G to C is 1. Okay. This is an experiment which he did in 1949. Okay. Till 1949, we have absolutely no idea what the structure of DNA looks like. You can see that this ratio is, it's there, it's close to 1. He did it for all other things also, but this number is the one which is close to 1. Okay. This is one level of information. What is it going to be useful? It is going to be able to tell us later on that the reason this is so is because A will pair with T and G will pair with C, and we will have a structure which forms a double helix. Okay. Last time I taught this course, or the year previous to it, I was asked a question of why is it not exactly 1, right? If you see the ratios here, they're never actually equal to 1 except in these two cases. That's just because he's using chromatography at that particular stage. These are crude methods. They will never exactly result into 1, but they do hover around 1. Okay. Now comes the math part. So Rosalind Franklin comes into picture. She's a crystallographer. She's actually, I think, a chemist turned crystallographer. So she's everybody's trying to understand what the structure of the DNA looks like. She has been photographing using X-ray structure of the DNA, and one such photograph looks like this. Right? It's very hard to understand actually what's going on. Right? There is some structure here, there is a lot of mass here, and if you zoom out, this is what it looks like. In the 2008 paper, which is a review paper trying to revisit what happened and how Watson and Crick used this photograph to deduce that this was a double helix, it shows that to be able to go from this to a double helix, you have to go through this Bessel's function math. Okay. So while this is a structure which a crystallographer or somebody who actually understands both crystallography and 3D structures and 2D structures can say that this must have come from a double helix, in 1949 or 1951, this was not so straightforward. But if you try to image this, and if you try to go from this structure to this structure, you require a transformation which uses math. Okay. So you need to be familiar with this math to be able to do this. But we'll not go there. What we want to study is A pairs with T, G pairs with C. It forms a double helix. This is interesting and important because it allows and it gives us a ladder for making double copies of it as well. Because remember, this is the genetic material. So if I have one cell where there is DNA, these cells have to divide to give life to new forms. And when this information has to be transferred again, it's being transferred at this level where you'll make again 2 copies of this DNA. The reason it's possible is because you have these 2 strands. There are certain enzymes which can unzip it. Think of this like a zip. You can unzip it. When you unzip, there is a direction 5 prime to 3 prime, which is essentially the direction of where your hydroxyl bonds are. So examples, this is adenine, this is thymine, guanine, and cytosine. Here is your 5 prime structure, which is once at one end this is the sugar phosphate backbone. One end of it is 5 prime, the other end of it is 3 prime, and it runs the other direction, other direction in the other direction in its pairing bond, right? What is different here is this bond structure. So on this end, you see that for 2 things to bind, you need oxygen to form or a nitrogen to form a hydrogen bond. So this OH, which is your hydroxyl end, and this is your hydrogen end. The pairing happens here. So this becomes your 5 prime, this becomes your 3 prime. There is a hydrogen bond being formed. There are 3 hydrogen bonds being formed between G and C, and 2 hydrogen bonds formed between A and T. Okay, any questions here? This is just the biochemistry. DNA polymerase helps you untrue or unwind it. So, sorry, make the second copy. This won a Nobel Prize by Arthur Kornberg, and I think his son will discover another enzyme later on. DNA Pol1 becomes a template, allows you to make a copy of this particular template such that whenever you are extending, the extension happens to be in the 5 prime to 3 prime direction. This is determined by the structure of your hydroxyl bond here, right? The OH bond. Missing a figure actually. Yeah, so this part is this particular part here where you have, so the way this is read is onto the third carbon what gets attached. So this is hydrogen end. This can form a bond with your other OH, which can actually be cleaved, and that forms your H2O. I think it should be, yeah, this is here actually, sorry. This is where I was trying to look. So when you have to extend this particular part here can form a bond with H. This becomes your water and is cleaved, and you have an extension in this direction. Okay. Any questions about this part? Okay. So overall, what can happen is while the polymerase is supposed to make an exact copy, so that when I'm moving in this direction, I make a copy. I again make a bond here. This is 5 prime. This is 3 prime. This is 5 prime. This is 3 prime. If the extension is happening in this direction, as the polymerase is moving, it is supposed to make a complementary pair here. Sometimes what can happen is, even though I'm supposed to get an A, I might end up getting a second base or a wrong base. At this point, I've construed a mutation, right? Often what happens is there are 2 types of possible mutations. You can either go from an A to G or a C to T. These are called transitions. The difference between transitions and transversions is there is no ring change that happens in transition. So this is your A, this is your G. They have the same ring structure. In transversion, there is only one single ring. If you go from 2 rings to 1 ring, that's a transversion. If you go from 1 single ring to the other single or to the double ring, then that's a transition, right? Based on this arrows, you can see that there are more possibilities of a transversion happening, because I can go from A to T as well, which is again a transversion, because this is double ring, this is single ring. Why is that something which probably does not occur in the nature so much? Often in the nature, even though the possibilities of transversions are higher, we see more transitions. You see a lot more mutations which are either A to G or C to T, but not as many as A to T. Why would that happen? The number of possibilities here are larger, right? But when we observe this in the nature, we see more transitions. So either you will see an A to G or a G to A or a C to T or a T to C, but you won't see as many of these ones. Any thoughts? People who have studied this? BB 101, does it teach translation, transversion? Okay. Mm-hmm. Okay, so you're saying that this to this should be easier. Okay, so that's very, very close, right? It's a structural problem because when I have to have a different uh, benzene or different carbon ring being added here, I have to go through structural conformation, which often will incur a steric effect based on all sort of things that can go wrong in biochemistry, and that's why you don't see as many transitions. As many transversions. Transitions are more common. Okay, so we're getting closer, where now we know the DNA structure, we know how it makes a copy of itself. Once it makes a copy of itself, now if I want to actually make protein, I want it to be transcribed into RNA, and then RNA is the one which will get translated. In prokaryotes, this step is straightforward. You start from a copy of a DNA, part of that DNA will become transcript, so not the entire DNA will often make the RNA copy. Some part of it exists just because either you have to give stability or you have to control how much amount of RNA you're going to make. And then your protein in the case of RNA will be, bacteria or prokaryotes will be of similar length as compared to protein. The case, however, in eukaryotes is much more complicated, because while DNA mostly have this particular yellow thing, which is your actual transcript surrounded by regions which do not make RNA, we have that region, but we also have something called introns. This part which makes your RNA is called an exon. It's exonic in nature, whereas this part is intronic, right? So introns are something which are again there for a variety of reasons, but we don't need that to be transcribed before we start to make protein. So the first step that happens is you start from this particular part of the DNA and you make what's called a premature transcript, which is a mix of, which is essentially very similar to what happens in bacteria, except that some parts of it are not really required for making the protein, and which in this case happens to be this yellow region. These are introns which will be spliced out before they get transcribed, as they get translated into as they get transcribed into the mature RNA or the mRNA. One difference here is everything here is happening in the cytoplasm because there's no nucleus. In this case, the transcription begins in the nucleus. The first step will happen inside the nucleus, and as the mRNA gets formed, which is the mature form of this particular transcript, it will get exported and then will be translated into protein, which happens in the cytoplasm, right? Which is what you were mentioning, for example. Okay, any questions about this? Okay, the reason it's a bit complicated in eukaryotes is because of this particular part, which is we have an initiation codon which is required. What is a codon? It's a 3 nucleotide letter, right? So you need AUG is your start codon, right? And it'll become clear how actually people decoded. That was also a math problem. So you need a start codon and you need a stop signal to tell it that I need a protein to be translated from here to here. Everything else is kind of like a controlling region. This is called an untranslated region, 5' UTR. It is located towards the 5', it's an untranslated region. This is located towards 3', it's an untranslated region. In eukaryotes, the reason we have so much complexity in terms of our proteins is because one protein or one gene can give rise to multiple mature RNAs. These are called different isoforms. So in this case, there are 1, 2, 3, 4, 5 isoforms which are possible. The reason it's possible is because, so first of all, actually this example is slightly different. Here, in certain cases, the transcription also needs to begin at certain point, right? So this is my DNA. This is, at some points, I can decide where my transcription begins. So this is called the transcription start site, TSS. I can have a copy here of the RNA. So I can either start here, or I can start here, or I can start here. It depends on the kind of sequences which are available. It depends often on the kind of complexes that will come and bind here. So for now, it's not very important in terms of what comes and binds here, but broadly what is possible is, because of the fact that you start at a different location, you can end up making a different mature copy of your RNA, right? It will be a slightly different protein, but coming from the same region in your DNA. This is your DNA. Part of it is exon, part of it is intron, and depending on where you start, you can make a different copy of the mRNA. What is also possible is you can have a different sort of splicing. So even though there are 6 exons in this particular case, because when I actually synthesize it, when I synthesize the pre-mRNA, this is your DNA. This is a gene called calcitonin or CGRP. Depending on what I retain, whether I retain exon 1, exon 2, exon 3 along with exon 4, or I retain exon 1, 2, 3, and 5, 6, I can make 2 different copies of your protein. And this is what happens in your body. This is found in thyroid. This is found in your nervous tissue. Okay? Same DNA locations, same gene, but 2 different isoforms. An isoform is 2 different proteins coming from the same gene. Yes. Yeah, so that's why when I started the lecture, I said, gene broadly is protein that is somehow going to form your functional part of your body. But you're right, the gene function, the definition of gene is a very contextual thing that you define, right? So in this case, for me, this is the gene and these are isoforms, but you could also call that this is one gene and this is another gene, because they're actually resulting in 2 different proteins. Yeah. Mm-hmm. So even for humans, we don't actually have that cap, right? So that's why I said that while it's 25,000, tomorrow maybe it will be 27,000. Tomorrow, day after tomorrow, it'll be 30,000. Because so far we have been able to catalog and observe this, right? So once you observe this, what do you do actually, right? Like you either you will do proteomics, you'll put this somewhere and you'll characterize that this corresponds to a spectrum which corresponds to this long of a peptide, and hence this isoform is observable, and it's possible that it came from this particular gene. So there is a series of experiments which people can do for isoform. You can actually do a full length PCR and see whether that entire isoform exists or not. So you'll have to do it characterization step by step, but in the last 26 years, we have been able to characterize a lot of isoforms and assume that they will also form proteins at some point. But characterizing isoforms is easy because of the throughput that we get, which we'll talk about a few lectures later. Okay. Mm-hmm. Yeah. So you can do that because again, during the course of evolution, so this is a bit of a mess, but let's do it there. So during the course of evolution, often what happens, so think about it, like if an organism has to evolve, right? So if this organism has to evolve,